Bien sûr, on est en live. Hola, buenos días a todos y todas. Gracias por estar en el último día del seminario de inmunobiofísica, donde tuvimos excelentes colaboraciones, participantes del extranjero y del poli. Hoy en nuestro último día tenemos a Jana y a Pierre, que van a finalizar con excelentes pláticas. Gracias por conectarse. Y Pierre nos va a presentar a Jana.
Hola a todas y todos. Voy a hacer como los últimos días, cambiar el lenguaje directamente. So I would just introduce you to Jana. Jana El Husseiny is a PhD student that we share but is shared between 3 different PIs. So she's suffering a lot. The first one is Aurélien Dumaître, who is an assistant professor in microbiology in pharmacy. Julien, who you met 2 days ago for the talk about micropipettes and cell mechanics, and myself. And she's working on unraveling the impact of mechanical properties of parasites and the implications of these mechanics in the dissemination and also the protection for us against these parasites. So she does very funny things. You will see it's amazing the amount of things that she does. So Jana, you can start.
Okay, hello. Thank you, Pierre, for the introduction. And thank you, Tatiana. And so today I'm going to be presenting my work, which is Unraveling Morphological and Biochemical Response to Harsh Environmental Condition in Coccidian oocysts. So what are coccidia? Coccidia are intracellular protozoan parasites that infect the intestinal cells of humans as well as animals. And these parasites are transmitted using fecal oral route. So whenever it goes and ingested by the human, the oocyst is going to correlate and the spores is going to go to invade the intestinal tract, and where it's going to invade the the intestinal cells, and to undergo sexual and asexual reproduction before being shed again in the environment, and then it's gonna spread the infection. These oocysts are highly resistant. They allow them to persist in the environment for long periods. And it causes coccidiosis, especially in young animals, poultry, and immunocompromised humans. So there is many genera of coccidia. One of the most known is Toxoplasma gondii, which is causing mild to asymptomatic for healthy patients. However, it causes symptomatics whenever it's immunocompromised, and it causes intestinal disease. However, it is differ in host specificity. In our project, we are working on Eimeria model, which is non-pathogenic for a human, and it was studied, easy to maintain under laboratory condition, and it has a well-characterized life cycle and host specificity. This Eimeria are shed in the environment, and it can be transmitted in the soil, water, and food. And these oocyst form are environmentally resistant form that enables the transmission and survival outside the host. It is composed of a bilayered oocyst wall, where we have on the inside 4 sporocysts. With an inside, there's 2 sporozoids that are responsible for invading the intestinal tract. As well, we have this polar granule, which is in the interior part of the oocyst. Going back to the oocyst wall, it is around 100 nanometer to 1.5 micrometer thick, and it is composed of a thin outer layer and a thicker inner layer. This wall is composed of acid-fast lipids, glycoprotein, and beta-glucan fibrils, as well as dityrosine links that are responsible for the autofluorescence of the oocyst wall. Going back to the life cycle of these parasites, it can be shed in the environment, then afterwards it goes inside the host and it invades the intestinal tract. So the stressors can be affected this oocyst, going from external stressors like temperature, disinfectant, ozone, pH, or desiccation, UV radiation, or others inside the host, like digestive factors and host immune cells. So first, we wanted to start with some disinfectant treatments that have been done before on Toxoplasma gondii. So they have seen that the bleach is decapping the outer layer of the oocyst wall in toxo, and the heat is inactivating them. So we wanted to test these conditions on our model to see if there is any difference. So we applied this on two different Eimeria, which is different in the pathogenic level of pathogenicity and the infection site inside of the intestinal tract. So at first, we did just look at the morphological characteristics, and we wanted to see if there is a difference. If we look here on bright field and autofluorescence photos, we cannot see any big difference. However, we did transmission electron microscopy, and if we go on the Eimeria acervillana, we can see clearly here in the untreated condition that we have this outer layer as well as the inner layer. However, in the bleached condition, we just have one layer, which is the inner. So the bleach was able to decap the outer layer. In the heated condition, we can see that we have both layers. We looked as well on Eimeria tenella, and we have seen the same pattern. In the untreated and the heated condition, we have both inner and outer layer. However, in the bleached condition, we have just the inner layer. So as a conclusion here, we have that the bleach was able to decap the outer layer of the oocyst wall. However, untreated and heat-treated oocysts retain both outer and inner layers. We then wanted to quantify these data, and to do so, we have created our own model using Stardust, and we were able to segment these parasites and to automatically detect the apparent area of the oocyst. So we run this analysis on both Eimeria acervillana and Eimeria tenella, and we can see, like within the treatments, we have this decrease in the area. We did as well automatize to be able to measure the intensity, the mean intensity of the autofluorescence of the wall by doing a donut shape that we're able to recuperate just the wall of the parasites. And we did the difference between all the treatments. And we can see clearly here that the bleach is reducing by half the values of the untreated condition of autofluorescence intensity. However, the autofluorescence of the heat didn't affect much. So this is coherent with what we found in the transmission electron microscopy, where the bleach had decapped the outer layer. So we are losing these dityrosine links that are responsible of the autofluorescence intensity. So these bleach were able to modify the wall autofluorescence. However, for the untreated and the heat, we have around the same values of autofluorescence intensity. To go further, we wanted to measure the mechanics of this wall. And to do so, we have used the technique that Julian talked about last Monday. And this technique is by using two micromanipulators, where we have the first one is holding a parasite, and we have the other micromanipulator, which is handling the indenture. So the micropipette are homemade fabricated that aspirate the oocyst at a specific aspiration, and it goes under constant speed towards a microindenter, which we already calibrated, and we know its bending stiffness. So we can recuperate the force required to rupture the force of this wall. So here what it looks under is a microscope. We have the contact of the parasite, rupture, then we have second one, and rupture of the second wall of the oocyst. So using Python code, we were able to detect all the proximal wall values, and we were able to plot it on the different conditions of Eimeria that had been undergone. And if we look on these data, we can see that Eimeria acervulana showed a decrease in the rupture force in the heated condition. However, if we look on the Eimeria tenella, we have non-significant difference between them, but we have this dispersion in this value. So we went and we looked on the orientation of these parasites because, as mentioned earlier, these parasites have a polar granule which is on the interior part of the parasites. So we classified our data based on the orientation where the indenter is piercing. So here we have the top orientation, and here we have the side and the bottom. So we plotted our data, and we have seen that the side and bottom are always have more force rupture required than the top in all the conditions. So we can see that the heat is decreasing the rupture force. However, it's particularly increasing the fragility in the top region. And we have seen another pattern where whenever we indent these parasites to rupture it, we have this piercing in all the conditions. But we have seen that sometimes whenever we indent with a microindenter, it doesn't pierce the wall, however, it's exposed from the top region. So we wanted to see where we are seeing specifically this pattern, and it had been seen in the heated condition, specifically in Eimeria acervulana. So this is another thing that heat is really fragilizing the oocyst, and mostly in the top region of these parasites, where the polar granule is situated. Then we wanted to see the porosity of this oocyst wall because, like we saw the morphological and structural difference, we have seen the mechanical, but we wanted to see if these chemicals are able to penetrate and to enter inside of the oocyst. So we used three different lectin staining, which we know that they're gonna stain the outer or the inner layer of the parasites, and these lectin is differing on the assembly type and the molecular weight, where we have WGA and MPL with small molecular weight around 40. However, the PNA is larger and with different assembly. So we, based on the literature, we can see that the MPL is staining the inner layer of the oocyst, so WGA, on the outside, and the PNA, we didn't find really where it's gonna bind. So we did flow cytometry experiments, and just looking on the autofluorescence and bright field photos that we have obtained, we can see that looking on the autofluorescence in UV or FETC, it's the autofluorescence of the wall. The TRITC is where we have the staining of these parasites staining of these lectins, because they are coupled to rhodomin. And we can see that in the bleach and heated treatment, we have little bit staining in some of these lectins. So we pulled the data from flow cytometry, and if we look first on MPL, we can see that the untreated and heat showed a positive staining. However, the most important was the bleach, where it was really significant significant for the MPL. And if we look on the WGA, we have seen the same pattern. However, for the PNA, we weren't able to see where it really binds. So for MPL and WGA, we can see that it can maybe penetrate inside and bind on the inner layer, or whenever we decap with the bleach and we remove this outer layer, these lectin were easy access to the inner layer where they're able to bind. So these two lectins were able to bind on the inner layer of these parasites. However, the question was for the PNA, why it's not binding on this oocyst. So we went and we did another experiments where we explode these parasites, and we have seen that in the unstained parasites, we don't have any autofluorescence in the trichophyllid. However, when we stain with the PNA, we can see that there is a positive staining of PNA. So the PNA, because of its molecular weight, weren't able to access the inside of the parasite. We were obliged to open it up to be able to stain the inside. So it gives us more information about the penetration of these lectins inside. So PNA does not seem to penetrate the oocyst wall under normal conditions. However, binding is stored on the inner layer, indicating internal localization of its target, which means not all disinfectant can be able to enter inside of this wall because of the different permeability of the different oocyst walls. Then we wanted to start exploring another disinfectant treatment, which is in collaboration with Actalia, a food industry that are using the ozone treatment as an oxidizing agent to be able to disinfect surfaces, water, and food products. So we wanted to see if there's an effect of the ozone on our parasites. So at first, we looked on our bright field and autofluorescence, and we can see clearly that we have something different in the autofluorescence of the wall. So we quantify them, and we can see that we have a small reduction in the apparent oocyst area. However, we have a significant decrease in the autofluorescence of the wall of the oocyst. So we are losing component, we are losing these dityrosine links that are responsible of this autofluorescence. We done indentation experiment as well, and we have seen that the ozone is fragilizing the oocyst wall, and especially in the top region of this oocyst. But afterwards, when we saw the morphological characteristic, the mechanical one, as well as a porosity, we wanted to see if these parasites are still infectious or not. And to do so we did bioassays experiments where we inoculated the chicken for 7 days, and then we have performed autopsy and we recuperated the lesion of the intestinal tissue, as well as the oocysts collected after 7 days. And we did that on different treatments, the control, the bleach, the heat, as well as the ozone. And as you can see here, we have a positive number of oocysts after 7 days of inoculation in the control. And in the bleach, we have reduced in the number of oocysts as well as ozone. However, for the heat, we didn't see any of oocysts found in the intestinal tract. So we can conclude that the bleach and ozone partially reduce the viability and the infectivity of the oocyst. However, the heat treatment is significantly decreasing the parasite viability. We didn't stop here. We looked on the disinfectants that are used to kill these parasites or to eliminate them in the environment. And we looked as well on the osmotic shock that can be exposed the oocyst since it's transmitted in the water. So we started at first by putting these parasites in highly saline solution. So put it at PBS 10x for 24 hours. But then we dilute it again, and we put it again on PBS 1x, which is the normal conditions where we preserve the parasites. So if we look here before on the bright field images, we can see that we have this buckling effect, this indentation on the oocyst. So we performed our quantitative analysis, and we didn't see any difference on the level of the apparent area of this oocyst. But if you look on the autofluorescence as well, there was no effect. We looked as well on indentation on the different osmotic concentration, and we can see that was no big difference between these difference, no significant effect. So also the buckling was observed on the oocyst wall, it did not affect the rupture force. Then we looked deeper in our analysis, and here is what we obtained. So here is a normal condition in PBS1X where we are indenting these parasites. And this is the rupture force curve that we obtain. So usually we have this as a baseline, and this is a T0, which is the contact time between the indenter and the oocyst. Then we have the forced rise, which is directly afterwards until the rupture of the oocyst. We looked on one of our video with PBS10X, and we can see that this is the curve obtained, and this is the T0, which is the contact time. However, we have a delay before the increase of the force of the force to rupture these parasites. So we did it on all of the oocysts that have been in both conditions, and we have observed that in PBS10X and then afterwards when putting it back in PBS1X, we have this delay of response before really increasing of the rupture. So although the rupture force was not affected, a response delay was observed, which is giving us more information about the elasticity of this wall. And if we look here, we have this buckling on one side of the parasite. So we wanted to see if it's more affecting one orientation based on the other, as we can see, as we have seen in the other conditions where the top is more fragile. So we looked on all our data, and we have seen that this buckling is either on the top, either the side, or either the bottom. We did our count, and we have seen that they are evenly affecting their different orientation. But we wanted to see if there's an effect on the rupture force on buckled oocysts versus the intact one. And we can see that just when after putting them again in PBS1X, we can see that there's a difference. So here, for the proximal wall between PBS10X, there was no such big difference between the two conditions. However, when we have the PBS1X, we have this difference between the intact and the buckled. So this is giving us more information about the elasticity of this parasite, as well as giving us that the buckled walls appeared more fragile than the intact one. Then, after putting them in high saline solution, we put them in water to see if there's an effect. So as well, we put them in water for 24 hours, and then we performed experiments. And as you can see, there was no this buckling effect on the wall, which is normal because the water is getting inside instead of getting outside of the wall of the parasites. So we checked the area, and we have seen the increase in the apparent area as well as the autofluorescence intensity of the wall, which is like the water maybe is entering inside of this parasite, as well as there is some components on the wall which is changing to have this dityrosine links, giving more autofluorescence than in PBS1X. We did indentation, and we didn't see any difference in the rupture force between PBS1X and water, but we have the top, which is more fragile in both conditions. So the water is increasing the apparent area of these parasites as well as its autofluorescence without any significant effect on the rupture force. So we tackled some of the external stressors that are affecting the oocyst wall in the environment, going from disinfectant as well as ozone treatment, which is one of the disinfectant, as well osmotic shock. But we wanted to see as well if there's any effect of the digestive factors inside of the host. So we started to see if there's any effect. To do so, we have done experiments on Eimeria acervillana with using trypsin, bill salt, as well as excystation medium, which is a combination of the trypsin and bill salt, and to see if there's any effect on the structural morphological characteristics, as well as the mechanical characteristics of the oocyst wall. And interestingly, we have seen that the area of the oocyst is increasing within this treatment. However, the autofluorescence of the wall wasn't affected. And we performed indentation experiment as well. We have seen that there was no significant effect within the trypsin or excystation medium, with little effect on the bill salt, as well as the top. It's fragile in all conditions. So exposing these parasites to digestive factors had led to an increase in the oocyst area, but without significantly affecting neither the autofluorescence, neither the rupture force of the oocyst. So as a conclusion, we have seen that these Eimeria oocysts are highly resistant to environmental and digestive stressors due to their thick walls. So we started to do wall structure, morphology, and mechanics based on two species, which is Eimeria tenella and Eimeria acervillana. We have seen as well the effect of the orientation of the parasites, where they have this outer layer, which is more fragile, and especially when heating this parasite, which is killing them. And we have seen as well that the outer layer is more sensitive to chemical stressors, where it's decapped using bleach as well within the ozone treatments. So this was the conclusion that we have seen in the external stressors. And within inside the host, we started with these digestive factors to see if there's any effect, but for the moment, no effect on the Eimeria acervillana, but maybe with the dynamics, we can see what's happening further in time as we are incubating them just for 10 minutes, and to see what's gonna happen further in time, as well whenever we put these parasites in contact with host immune cells, like macrophages, neutrophil, or dendritic cells. So the perspective of this project is still to study the dynamics of the digestive factors, then to explore these mechanical links between the structure, the morphology, the mechanical stressors, the mechanical strands, and the infectivity of these parasites. And to finish up with the recognition of the parasite with the immune cells to see if there is a difference in the response between the oocyst versus the sporocyst inside of the oocyst. And I would like to thank my supervisors, especially Aurélien, Pierre-Henri, and Julien, and all our lab members. And I would like to thank you as well.
Thank you, Jana. Tatiana maybe just escaped for a few seconds. No, she's there. Because she's the one who has the questions online.
Sorry, Jana, thank you so much for the great talk.
Thank you.
Some beautiful images and techniques. And I will ask now to the audience if they have some questions. Otherwise, I will do some questions. But okay. First, well, one question I will try to say. Heat significantly reduces oocytes area in the acervillana, but not in tenella. Do you have evidence that this morphological difference reflects species-specific difference in wall composition or in proteins cross-linking, or what do you think?
We didn't tackle exactly the molecular level of the oocyst wall. However, yes, we have seen that it's species dependent in morphological way as well as a mechanical way, because like in Eimeria tenella, we didn't see any fragilizing as much as we have seen in Eimeria cervellana. So it is really related to the biomechanical molecular level. So this is a point that we wanted to understand more to see if it's affecting the molecular level or not.
Okay, then in the lectin staining, MPL and WGA staining increased after certain treatments, suggesting inner layer exposure or penetration. Do you interpret this as true permeabilization of the wall, or rather the unmasking of previous hidden epitopes? Do you think it's a permeabilization of the wall or other thing? Or
yes, I think it is because of the permeability of this wall, because whenever we decap this outer layer, we can see that, it is here. So first, in the untreated and the heat, we have these lectins that it may be non-specific to one layer of the parasite. It may be as well binding on the outer layer. But we have seen that whenever we decap this outer layer, the lectin is able to bind the inner layer. And I think it is a matter of permeability, because with the PNA, whenever we open them up, they were able to stain the inside of the inner layer. So I think it's a question more of permeability of these lectins.
Okay. And then in the osmotic stress with water, hypoosmotic shock increased area and autofluorescence, but did not affect rupture force.
No.
Have you explored whether these treatments alter elastic or viscoelastic properties beyond rupture strength? Or
yes, because we have did some experiments using different speeds to be able to see if we are able to rupture the force and to have the viscoelastic properties, and we didn't see any difference. So it is not a really viscoelastic properties. It's more likely it's an elastic of the wall.
Okay.
If I may add a comment, the wall is very thin, so measuring something viscoelastic on the wall is very difficult. We try to see whether it has an influence on the distance between the peaks, because this is where you travel through the oocyst, where you visit the internal system. But here the situation is mechanically very complex. You have a fluid plus moving objects. So we tried with Julien to model this, and this is very difficult to gain any real feeling of what's happening inside. That was just a comment.
Thanks both. And we have two more, and we let you go. So first, digestive factors expand oocyst area, but did not weaken the wall. Does this support the idea that excystation depends on localized weakening zones rather than global mechanic softening? Or
so for the moment, we have seen that this area is increasing. So it may be leading that because of these digestive factor are maybe able to penetrate inside and be able to start the excystation process. So it may be due to that. So your question was about, sorry, I forgot the...
Either it's a global mechanical softening or a localized weakening.
Yeah, I think it is more likely to be localized. So maybe we have this polar granule where it's really the rupture inside of the digestive system, and then afterwards these digestive factors are able to excystate parasites. Yeah.
Okay, and the last one, bleach and ozone reduce infectivity only partially, while heat almost eliminated. Do you think the main determinant of in infectivity is damage to the wall or direct damage to the sporocytes themselves?
I think it is more maybe, no, it's affecting maybe the whole parasite. So it's affecting, whenever we affect the outer layer, we don't know what is happening inside. So if the products were able to penetrate and to kill these parasites, so it may be due to the fact that the population, some of them were more affected than the others. And this is because of the fragilizing of the wall. So maybe it's due to that.
Okay, well, thank you so much, Jana, to having you here. Of course, we hope you can come to Mexico to show us all these beautiful techniques and the knowledge you have.
Here.
Visit us.
Thank you
Jana.
for
And have a nice evening.
See you.
Thank you.
Bye, Jana.
Bye. And can I watch your presentation, Pierre?
Yeah. You can stay.
So I'm gonna stay connected.
Can you please stop sharing so I can share my own?
Yes.
Yeah. So for the audience, our last speaker, who is going to close this wonderful seminar we have during the last 4 days, is Pierre-Henri. He came from Marseille, Lumini, and then his lab is a laboratory. Ay, no, ¿y por qué estoy hablando en inglés? Perdonen todos. Su laboratorio es un laboratorio de adhesión e inflamación en Marsella. Es un amigo, es un colaborador de nosotros. Viene cada año al laboratorio, así que quien quiera venir a platicar con él, a sacarle la información, a exprimirle todo su conocimiento, es bienvenido a venir al laboratorio. Cada año tenemos estos seminarios en donde aprendemos de lo que están haciendo y tratamos de colaborar con muchas técnicas y conocimientos que tienen. Entonces son bienvenidos a conocerlo directamente en el laboratorio. Va a estar aquí todavía hoy y mañana. Y el año que viene seguramente. Bueno, a inicios de año también va a estar aquí y los invitamos a que vengan. Entonces es nuestra última plática de cierre magistral. Muchas gracias, Pia, por estar con nosotros, por este esfuerzo de colaboración y porque sigamos trabajando.
Vamos, okay, maybe direct element go in English. Thank you, Tatiana, for the introduction. As Tatiana mentioned, this is a long-lasting collaboration that we have between UNAM, the Landsbioaudit, and our lab, which is in France, which is supported by the CNRS. I don't know if you can see my mouse, but the CNRS is this small stuff. It's the National Center for Science. So today, I will try to bridge a bit some different aspects that you've been hearing in the last few days about the immune system, but the biophysics of the immune systems, introducing you to different techniques that may shed light on what is going when the T cell encounters its target. And what happens is on this very, very nice watercolor painting, the T cell, which is at the bottom, I think, has to recognize the problem that comes from the top cell just by finding this small red dot that is in the middle of the image. So this is a very delicate system. So T cell lymphocytes, they are those cells who are scanning your body and try to figure out these very small, tiny infection markers that are expressed by your antigen-presenting cells that we call APC. They have to read the antigen and react accordingly. How does it look like when you do that on the Petri dish? This is this very famous and very beautiful movie where you can see the red cell is the T cell scanning very actively the antigen-presenting cells. So a little bit like a blind person trying to figure out what's happening in the microenvironment. And then it does that through a very particular receptor that is called the T cell receptor, or TCR, that is reading another molecule that is a major complex of the major histocompatibility complex, MHC, of different types, depending on the cell. I won't go into the details of the biology there. I think some of you may know more than I do. And they try to figure out this antigen. And this triggers the response of the T cell. It has to be fast because you don't want the target to escape, reliable, you don't want to make an error, and sensitive because the information will be very little presented on the stuff. I think it's 1 over 1,000 MHC molecules that represent a peptide that have to be recognized for saying that it's a danger. The others will be bearing the information that everything is fine, it's yourself, and it's not infected. Not only cells can understand the biochemistry, but they can organize, as Kaya mentioned, at the membrane, their molecules. You will have this kind of immune synapse that was touched by Gaurav yesterday or 2 days ago. In fact, this immune synapse is the reorganization of the receptors at the membrane. In the central part, you will have the recognition molecules, such as the TCR and APC, plus co-receptors. At the border, everything will be sealed by adhesion molecules from the integrin family to make a tight seal. And then you will expel the glycocalyx, another molecule that was discussed by Kaya yesterday, on the outside. And this will generate the possibility of integrating the signal from the molecular to the cellular level. But there's a lot of mysteries about this integration, how you can get at the same time sensitive, reliable, and fast. Because this is, for physicists, very incredible that the system is able to get those 3 parameters optimized at the same time. So T-cells do that also by integrating physical cues. And what kind of physical cues? Some molecules, such as integrins, but also the T-cell receptor, can feel the forces. And there's a lot of work in our lab, in particular by Philippe Robert, who is a medical doctor, but also a researcher in the biophysics side of the T-cell, to understand how these molecules feel at the single molecule level, the forces. Maybe they change conformation, as Marie mentioned 2 days ago, or some other properties. There could be a catch bond. Catch bond is a bond that lasts longer when under tension compared to slip bond that will simply dissociate. So potentially having complex behaviors in the bonds could help the cell to integrate that. Not only they feel the forces at the single molecule level, but they respond to substrate mechanics. That means they can feel it as a wall. Like if you were touching something with the finger and then feeling with the hands or feeling with your body, the mechanics of the surrounding media. And they use also forces to perform the action. If you want to kill a cell, you want to make a hole in it. To make a hole in it, it's easier if you pull on it to make it more tense. This is what happened for killing T cells. And this is a very great job made by great work made by Julien, that you just heard. It's also true for other lymphocytes. You've seen yesterday B cells from Paolo, but Julien also mentioned neutrophiles, and also I can mention macrophages who do the same thing. So this is a common feature of all the immune cells. So that we call that immunobiophysics. At the same time, antigen-presenting cells, they can modulate their properties in response to what they feel. That means whether they have been contacted by a T cell, but also whether they are infected or not. And this has been demonstrated in 2015 by a very elegant study by Buffy et al., where they shown that immune cells have different mechanical properties, but these properties are changing depending on the moment where they are in the inflammatory process. In particular, DC, they are very soft, then they become stiffer when they become primed. So all of that tells you that this stuff, you need mechanobiology to decipher what happens in the immune systems. And in the lab, and collaborators such as Kaya, Laurent, who you met last year, Philippe, who is our medical doctor, and others in the lab, they have worked on that field to try to understand and unravel this mechanical sensitivity and mechanical bias that the system has from single molecule to single cell level. Today, I will simply touch a couple of them at different scales with different tools to show you how we try to streamline this interaction and streamline this knowledge and questions about the physics and the biology in these immune systems from the single molecule scale to antigen T-cell presentation interactions. And you will touch different techniques such as atomic force microscopy, soft substrates that you do in NANS-Biodit, micropatterning. This is the master of Amrani. T-cell APC interactions. This is the master of Jana here also. Optical tweezers and traction force microscopy that you heard yesterday with Paolo. So the first thing is, what is atomic force microscopy? It's a very simple spring that can feel very little forces. It relies on the cantilever that makes a couple of 100 micrometers. You detect the motion by pointing a laser on it. And then what you do is simply approach the system, press on it, so the force increases, pull on it. If there's no adhesion, you come back to the baseline. if there is adhesion, you will have jumps like that will reveal that some adhesion. And the magnitude of the jump, once everything is calibrated, as Jana mentioned really recent, just before, when the stuff is calibrated, you can extract the forces. And believe me, this can be sensitive to the single molecule level. I won't demonstrate it, but in fact, this is true. So what did we do? The question was, is the T cell reading the rupture forces of the TCR PMHC interaction? And just to emphasize the signal of the peptide, is it stronger? Is the force strong? Is the force stronger or weaker when the peptide has to be detected? So we did that by having T cells, resting T cells, gently adhere on a substrate and presenting a cantilever decorated with very few PHC molecules where we can tune the peptide. What we did, we tuned the peptide. We changed the peptide from no peptide, non-activating peptide, super activating peptide. Very often you have this contact, and we went for very, very gentle forces just to be able to get into the single molecule range. This we can discuss later on, but you have many ways of reaching this regime. And very often we had no adhesion. And sometimes we get some small peaks, but we're on the 20 piconewton range. 20 piconewton range is very weak. As Kaya mentioned yesterday, it's only a couple of times the energy of simply of Brownian motion. And it's very low compared to adhesion molecules. Adhesion molecules, they are more on the 100 or to several hundred more piconewtons per molecule. So what we've seen is we varied the peptide and we varied also the presence of a co-receptor that should be increasing the adhesion strength of the stuff. Very disappointingly, we found that no effect on the force. However, by looking at the frequency of our adhesion, we saw that this was responding to the peptide. That means that our experiments were indeed specific to the peptide. So in our condition, force was not seen to be different, but multiple adhesion were more frequent, and even more frequent when you had the co-receptor, which is normal. You have one molecule interacting and the second one coming here. You may detach the two to break the bond. So our conclusion was that in our experiments, the co-receptor, in fact, was helping the system to not only be specific, but to have the time to really orient properly. The geometry of the encounter of a TCR and PHC molecule is depicted here in A on the right side. It's 45 degrees. It's only on a couple of amino acids, so it's very complicated. So introducing this receptor makes the system look like a time lock for having this interaction. And we didn't have the resolution to see this force. And what happened is 5 years later, people have seen that force is not the good readout, but it's the lifetime of the bond under a given readout that shows that this TCR is potentially a cache bond, and the range of the force where this sensitivity is optimal is 10 piconewton. And 10 piconewton was the noise of our technique. So sometimes you don't have to forget that you have noise in your experiments, and that noise may be limiting yourself to what you can read. Recently, people have proposed that CD8 could act as a time lock, which is compatible with what we just proposed in 2011. So as I said, AFM was not a good technique for that, but at least in our experiment, we've been clean, and so it took us 4 years to get that. And since we had it, we said, okay, let's do something else with the AFM. We try to play with the AFM as a way to control the contact moment between a T cell and an APC, an antigen presenting cell. So we use a model APC, that is something where you can transfect the molecule you want. You can add co-receptors, you can add additional molecules, and then you can have this TCR that will be highly specific of your T cell, and you can tune the peptide. And the idea was to use the AFM as a hand to bring the cell and contact it. And also what we wanted is to have cells, when adhere on the substrate, to be more homogeneous. So we try to pattern them, to get them with a given shape, and hoping that controlling the shape, let's say not spread, well spread, we will also control the state of mechanics, for example, the state of tension. So this is really a physicist's way of looking at the earliest moment under force of a T cell and an APC contact. The first thing was to immobilize the cells without activating them. These cells were hybridomas from mouse, and so we had to find ways of having zones of adhesions and some of non-adhesion. And we played with maybe 40 conditions and finally finding, and here you can see calcium fluxes. These are the records of calcium fluxes. In red, you have high calcium. In blue, no calcium. What you see is if you put the cells on the assay, they do not activate. So that's a good repulsive condition. If you put them on anti-CD3, which is an activating molecule, you activate them. The good positive control. If you put them on anti-CD45, which is blocking this glycocalyx, in fact, you don't activate them much. So it's a good candidate. And if you combine immobilization with anti-CD45 and put soluble anti-CD3, you can reactivate them. So that means that you can have cells non-activated but activable. In the word, you do not, let's say, repress them from activating by having them adhere. I just don't comment on the right side of the stuff. This is just the quantification for saying that the cells are immobile, stuck on the or stuck on the surface. They do not crawl on the surface like they would do for the, let's say, you can remember the one from Gorav a couple of days ago, but we're running here and there depending on the molecules that were on the substrate, and that we can reactivate them. And then we went for patterning. The problem we encountered is that patterning is sometimes irreproducible. So we had to, in fact, find the weakest point of our patterning system. We were producing ourselves the anti-CD45 antibodies, and they were a bit fragile. In the stamping method, you ink the stamp, you dry, and then you stamp, and then you complement the surface. And during this drying step, we had some problems. We maybe were unfolding slightly our antibodies, so we went for the reverse strategy. We were printing, in fact, the repulsive zone. Here you see bioattenuated BSA labeled with fluorescence reptavidin, and then we absorbed the anti-CD45 that was not labeled. And this is even good because you do not change the properties of the antibody by putting the fluorescent molecule. Then we had the cells sitting on that, and then they were sticking. Here is RICM images, just as what Gaurav presented yesterday. And you can see that cells were taking the shape of the pattern, and when confocaling it, you could see this is a typical image on the right, on the bottom right, that we had half spheres of cells with the nucleus a bit close to the surface. Having that, you can measure the mechanics, simply pressing with the atomic force microscopy. This is the similar system as what Julien presented. the cytoskeleton. It should not be when we pull the tube. Eta is kind of an effective viscoelasticity. It's the way it encompasses the transition from the cell to the tube. So that means all this viscous part. So when you have a breakage inside, you should have a spike in your curve. When simply you slide out the cytoskeleton, the tail of the molecule slides out of the cytoskeleton without breaking anything, you should have a very smooth transition. And this was modeled by our stuff. What we did is, in fact, to play on the type of molecules we're putting on. So we use, as a control, the integrin. We know that when an integrin is open, is very highly interacting with the cytoskeleton. When it's closed, it's weaker. We use the CD45 as another molecule, but where we don't know the interaction. We know it's corralled very well by the cytoskeleton, and we put also on the TCR complex. We use antibodies to have a strong outer bond to be able to break the inside bond. And what we saw, I just present here the results for K2 and K1. K2, no difference when we are putting with the different molecules. K1, no difference. That means that roughly the stuff that we have at the inside is the same. And then we introduced a variation of latranculin. Latranculin kills at very low dose, killing and perturbating the cytoskeleton. When we introduced that, we saw that K2 was always decreasing, which is good. We touch the cytoskeleton, so the entire cell mechanics is changed. But very interestingly, only some molecules were having a K1, that means the bridge inside the cell, modified by the perturbation of the cytoskeleton. That is, namely, the straight integrin that is strongly interacting with the cytoskeleton, and the CD3. So as a conclusion, the tuning the cytoskeleton and measuring those parameters by fitting our curves shown that our model proposing that you have a differential interaction with the cytoskeleton depending on the condition is right. So from that, we were able to bridge somehow the TCR to the mechanics and try to understand a bit what happens from the outside to the inside of the cell. And this introduces something that was long-lasting. It's a long-lasting question, and not even with this paper we fully resolve, I think, is what are the physical links between the TCR and the actin? From outside to inside and from inside to outside also. You have to see the two, because you could be pushing actin for simply having fingers to probe the structure, to probe the substrate, as Gaurav mentioned a couple of days ago. And in fine, the goal that we would like to have is to link what happens from the molecular level to the cellular level. Looking at the cytoskeleton is one way, and we wanted to find another way of doing this. And Paolo mentioned that traction force microscopy. Here I just put a panel with different techniques and all the techniques that have been used in immunobiophysics by many different groups from, let's say, from our lab, from labs in Netherlands, from labs in the States, and so on. And basically, we went for developing this traction force microscopy, that is in fact having the gel with beads inside that will serve as fiducial markers decorated with the given molecules you want, and then recording the traction and the contraction or the forces that you have on the gel. The first task was to make those gel reproducible. Why? When you want to do typical gels, the gels are typically between 1 or 5 kilopascal and 20 kilopascal. This is what you see for large cells. The recipes are very stable there. So this is fine. But if you want to look at what happens when a T cell meets a B cell, the B cell are 100 pascals. So basically that means that you have to go lower in gel, and the lower you go, the more problems you will have. Is the gel elastic? Is the gel viscoelastic? Is the gel reproducible? What is the porosity and so on? But you stabilize the problem, and we use AFM to map at the cell scale the elasticity and show that it was rather homogeneous. We went for many different situations, and we obtained the gels in the range of typically 400 pascal was the most stable part. So we use them, and first we measured the spreading by using fluorescences, and we saw roughly the same kind of trends for primary cells, for jocket cells that are the same cells that were used during the experiment with Kea on soft substrate. And we use also primary cells extracted from blood bags. And here, what we saw is that there was different spreading depending on the subtype of those cells. And this is the first time that someone works with many different types and look at this biophysical parameter. We did also the mechanics, and we saw that the stiffer the substrate is, the stiffer the cell becomes. It's also because of the spreading, so that's logical. But also what we noticed is that depending on the type of primary cells, their mechanical properties were also very varying. So that's also the first time we see that. These results were obtained for the Jurkat part by Farah Mustafa, who is now a postdoc in New York in Morgan News Lab, and the primary results also. And part of them were obtained by Jana, you just met during a master thesis. And then we went for distraction force microscopy for the earliest moment of the cell recognition, just like what Paolo presented yesterday. Cells are sedimenting onto the substrate. You have the beads at the initial position. When forces will be exerted, beads will move. You can track those beads. You can PIV them to get the displacement. You can reconstruct the force field. From that, you can extract the stress over time, because you will record a movie of the cell pushing and pulling on the stuff, and then you can measure the energy. You can measure the integrated energy as you do for example, for any kind of quantification to get a single parameter. What we saw is that when we have this anti-CD3 substrate, the anti-CD3 gels, the stress, that means the forces, were higher than on control antibodies, and the energy also. So that means that we were specific. And then what we observed is that depending on the not only depending on the substrate, here you have images taken from movies from on control substrate, but also on activating substrate. You have different patterns that are spatially and timely of this energy versus time. So mainly what we observed is that we had four different patterns: oscillating, no signal on control substrate, very little interaction; oscillating but higher signal on anti-CD3; intermediate signal that raise and falls; and sigmoid signal that are raising very strongly. And what we went for is to say, okay, is it a signature? Is there any information again in this kind of different patterns? And yes, you have it. In fact, what you have is you have very different patterns depending on the condition. Here on the between AGJ2A and anti-CD3, that's the first signature. But if you tune now the stiffness of the gel, going for this optimal condition that we've seen on the spreading experiment is 2, 3, 4 Ks, something like that, you see that you do not modulate much. In fact, you decrease a bit the stress, you decrease a bit the energy. That says that with these very small and very sensitive cells, you may lose some of them because you won't be able to detect them much. But also what you see is that the type of patterns, when you compare two experiments, let's say when you compare experiments made in the same condition with the same cells, or coming from the same blood bags, you don't have the same kind of time patterns. So it seems to be, these patterns seem to be sensitive to the external microenvironment, but also if you change a little bit the cell, if you translate the cell with things that could be either life hacked or put a marker in a membrane, you see that the forces and the energies are not that much different. What changes? These are the distribution of force patterns. So these force patterns are very sensitive to subtle things. And when you apply that to different families of primary T cells, such as, or either what we just seen is just Jurkat cells, that means model cells, you take PBMCs and differentiate them in naive, activated, and effector, memory, and you reactivate the memory and so on. Can you see differences? And the answer is we at least see the different patterns. This is the proof. We even see spreading cells or contractile cells. Spreading cells is very rare to see in literature at the moment. But what we see is that the forces and the magnitude of energies are different, varying depending on the cell subtype, but also the way they generate the forces. Naive stimulated go for intermediate. That means raising and letting go, raising and letting go. When you go for memory T cells that are not activated, that are not being stimulated, then you have the maximum speed and efficiency of response against time. This is not difficult to understand when you know that memory T cells have to be the fastest to answer because they already seen the problems they have to encounter. So you can really link that to the subtype and the activability of the subtype. which is very good. This was already said last year during the conference that I gave during the first version of the seminar, but since then we had some more questions. How to analyze space. And by having a discussion, by Keia having a discussion with Pierre Onseret, who is a theoretician in our lab, he told her about the Cauchy stress dipole tensor. So don't be afraid about that. It's a tensor that contains that you can calculate, that contains information about the orientation of the vectors. In fact, you can diagonalize it, and depending on the sign and variation of the terms, you can know whether there is an order and whether it's contractile or extensile. That means extensile or contractile forces in regard to the contact area of the cell. So we did this calculation. I have to say, Keia did this calculation, and we plotted the results. And what we observed on JERCAT is that the eigenvalues that are the signature of this orientation are strictly following or very closely following the energy. Here you can see intermediate signal, and you can see that it goes down. Sigmoid, it goes sigmoid and change the sign. Negative means like this, positive means like that. So what we've seen is that in fact we have a parallel between the type of time pattern, energy pattern, sorry, here, magnitude of energy patterns, and the spatial stress pattern. That means we have random for this active noise that is larger than the non-specific interaction. For intermittent, we don't have much of an organization, but we have for the sigmoidal two behaviors, in particular for memory cells, one where the cells were pushing and some where the cells were pulling. The pushing case is very difficult to understand, and we are still munching on that. But we propose different ways of looking at this. And then you have different scales at play at that. The first one for the active noise, the cell is simply touching with fingers. This has been seen in 2013 or not 2005 or something like that by Pierre Bongrand, who was the head of the lab. In fact, the cell is in fact sending fingers and poking like that. Poking will displace sometimes the bits vertically, and this is where the traction force microscopy per se is not good enough. We cannot see the out of focus forces. For the intermittent, your finger comes parallel to the substrate and you start to push or pull a little bit more. Then, when the signal is integrated, then the cell will either spread, that will be the extensile, or won't spread, contract. This is the contractile stuff. So in fact, by having those readouts, you may see how these kind of transitions in your population is made. And don't forget that each cell will be different, in a different energetic state, in different even activating state at the beginning. And the main question is, when you take a population of cells, what is the starting state? This is the start of our next project. We think that the starting state is metastable, mechanically speaking. That means that is, in fact, just waiting to be pushed a bit to get to the interaction. So as a conclusion, we have many different ways of looking at these different scales. But what lacks for this spreading and traction force microscopy experiment? This will be the start of the next PhD student. We will be, I think, beginning of next year. What is the difference coming from also some subset that we did not analyze? I mean, CD4 plus, that means helpers, or CD8 plus, that means killer cells. Do they have different patterns? And also, what is the relation of these patterns to calcium fluxes? We did not have much time to do that. And when we did that with Jana, we ran into some technical problems of detecting at the same time the beads, detecting the contraction, detecting the calcium fluxes, and we ran into the problem that Julien just talked about. The light we put for detecting the beads was simply stopping the activity of the T cell. So we have to find, it's not complicated. It's just like fine-tuning the system to get the two at the same time. And also, you have to detect the beads at one plane and detect the calcium on another plane. You have to be able to make two images at two planes. Technically, this is doable. It can be a bit tricky. All in all, the take-home message is that T cells and immune cells in general, they have a very physical and lively life. They are using forces to read the message. They integrate it at different levels, from single molecule to single cell. They use it for acting on their environment and, for example, to keep pathogen. So we can try to harness that. And to harness that, is what Gaurav presented. That means you could, using and knowing the mechanical properties of molecules, try to scale the answer of the cells for killing, for example, for NK, for T cells, for CD8 plus T cells, or even macrophages to do this, the ADCC and MCC strategies. Again, I would just thank all the persons involved in this work from very long. These two at the top left were my first students, and this is back from 2011, to my latest student, Jana, who is still in PhD with us. And with that, I will simply finish by 2 words. We organize a series of conferences about immunobiophysics. It's held in Nezouche, and it's at a crossroad between physics and biology. It's funded by the EMBO, the European Molecular Biology Organization, and it takes place in a physicist world, that is the School of Physics in Nezouche, close to Chamonix, close to Geneva in France. In April 2027, it will be the 4th edition. What is more interesting is that it's a mixed edition. That means you have a on-site 70%, but also you can follow the conference online with records and everything. And in parallel, we have this free seminar series, and there's one just now at the moment going on, where you can follow the latest of the immunobiophysics research. And I will finish by advertising the fact that in Marseille, we have this Century PhD program. Here, this is just the example of the one topic that I proposed for this year. You can apply. There's also Kea who has a topic there. Please go on Century Turing Center to see for the PhD program what is this. There's also a master program. It's very interesting, and it's open, really open for international students. So with that, I will thank you for your attention, and this is the way we can recompose a small painting that I like, using cells and beads.
Thank you, Pierre. I think that it was like a really good idea to have you at the end to pull together all the parts and mention what others say during the seminar. And we do have some questions, also a little bit integrative. So, how do T cells combine molecular level mechanics and with whole cell viscoelasticity adaptation to extract meaningful information during the very first seconds of antigen recognition?
So this is very difficult to answer. There's plenty of ways of looking at it. The first one is that you have the possibility of molecular local amplification systems, such as this kinetic sorting of molecules and also geometric sorting of the molecules at the membrane. To go beyond the membrane, you will have to take into account the fact that the signal can be integrated by the cytoskeleton and other signaling molecules. For the moment, there is no view of this kind of integration. And one of the propositions to try to solve this kind of force integration, we mentioned that yesterday with Paulo, is to integrate different ways of measuring at the same time. In fact, you could measure local, very molecular level forces by using DNA sensors, add traction force microscopy as a cell scale, and at the same time record calcium or any kind of phosphorylation on the back. This is the only way to cross the scale. The only problem is that you will need to model what happens. And the space and time are playing against you. They are in fact making the stuff really non-linear. So at the moment, it's a very hot topic of research, I have to say. So the answer is, this is a good question.
what's still blind to? How can we integrate all these together?
The point is to try to have systems, physiological relevance that we can control, and we can make measurements at different scales. This is basically the main issue that we always run. You have some very refined experiments at single molecular level with certain type of molecules. Some very refined imaging of structures on certain cells, on certain molecules in certain conditions. Difficulties to get information and to make it a continuum. It's good if you are able to do it across scale, set that control very well on substrate that are exactly the same. So you have to make possibly different bind them. This is the way I would see but I have to say that something I'm still struggling with. A lot of information exists since 1970 on the biophysics. Been studied like hell. Immunology is one. Lot of information already there. How do you do is to the lazy man approach. The lazy man approach is to leverage LLMs to try to figure out what were the conditions and experiments done. The idea was to ask to, in fact, in a system some articles we were interested in, so they can extract the experimental part, and then follow the different goals and recover the conditions, which cell, what molecule, which cell, what molecule. And if you experiment yourself with the same systems, or you try to combine already what has been already found, and you expand it, the cross scale is the key, from my point. So from one molecule to one cell, and then from one cell to one cell and its partner, from one cell to a new organoid. This would be the key to answer the different problems, taking into account the fact that you have mechanics, you may have this kind of fluorescence events and so on. It's very complicated, but that would be the key.
Bueno, una vez más, gracias a todos y todos los que están conectados. Gracias por haber estado en los seminarios estos días. Gracias, Pierre, por venir a visitarnos y empezar este seminario. El año que viene, les prometemos, vamos a tener también la parte práctica. Vamos a tratar de traer a los invitados a que personalmente den las pláticas y con ello puedan más, aprender más de su conocimiento, no solo teórico, sino práctico. Pero vamos a hacer un esfuerzo que cuesta dinero y eso es un problema. Pero muchas gracias a todas y todos.
Gracias.
Y cerramos el seminario.
Y nos vemos el año próximo. Ojalá en México.
Sí.
Hasta luego. Muchas gracias a todos.
Adiós.